Greetings, everyone, and welcome to session number 7 in the FBIS training series. In the last few sessions, we focused on the different kinds of dashboards for visualizing and exploring the different kinds of biodiversity data. And today we're going to be taking a deeper dive into how you can actually take that a step further and download information from the dashboards, whether it's graphs or more detailed data sets in the form of CSV files. So really just giving you the maximum power to access data through the FBIS platform. All right. So today we'll use a case study of the Rondegat River. It's a river system in the Western Cape where there's been quite a lot of conservation attention and scientific monitoring around freshwater fishes. So we'll use the search bar here to search for the river name. And just a reminder, in the search bar, you can search for site code or for a taxon or for a river name. And you can see the Rondegat River comes up, and that'll take us straight to the Rondegat in the Cederberg. And you can see here various different data points from the different monitoring efforts that have happened here. We can also then just take our lasso tool and maybe just hone in on a subset of these data by drawing around them and creating a polygon. We hit update search, then that gives us that data set for those sites. So we can see it's around about 1,500 records from about 21 sites, 117 taxa. So if we have a look at the sites overview, we can see that for these sites, there was actually a range of different kinds of data collected for fish, for invertebrates, and also for anurans. We can then drill down into the multi-site fish dashboard and visualize the data there. And we can see that this is quite an interesting set of sites because they were sampled as far back as 1967. So a really nice long timescale on this data set. And just a quick reminder of what these dashboards look like. For the multi-site dashboards, you can get you get a map of where your sites are, you get a quick summary of your filter history, and then you get some basic information about the records, a breakdown of some of the key attributes about the taxa recorded at that site, information on the most recent site visits, and then importantly, the occurrence data. And then on the right, we have most of the key data visualizations, quick summaries using pie charts by packaging the data according to some of the most important attributes: origin, endemism, conservation status, sampling method, and biotope breakdowns. And then the occurrences can be visualized over time. Packaged either just as occurrences themselves or separated out into the different taxa, different native versus non-native species, and different endemism and conservation categories. So the first thing I'm going to show you is just how to download a chart from this dashboard. And that might be of use if you're wanting to use this chart just as it is in a report, for example. So just a reminder that every time you see this little orange button with an arrow, that means you can download a picture of the graph, in this case of the table, in this case of the map. For the map, you may actually just want to zoom in before downloading it, so you can, for example, get a nice framing of where your sites are relative to the Clanwilliam Dam and Clanwilliam itself. All right. So if we wanted to download this, this chart of occurrence records over time, we would hit the little download button, and that brings up a little pop-up that asks you what is the purpose of your download. So you might be using it for a scientific article, for a report or a thesis. You might be using it in a proposal or a presentation, or no specific use, which is what I'm going to say today because this is actually just a test. However, if you were using it for a report, in the notes section, you would enter the details about that report, which is really useful feedback for the FBIS platform as well. Then, once you've filled that out, you hit the download button, and that gives you back an SVG file of your chart. This is a nice high-quality file that can then be exported as a PDF, or it can also just be saved as an image file on your desktop as well. All right. Back to our dashboard. The second thing I'd like to show you is how to download the raw data. So all of the raw occurrence records from your query from these sites in the Rondeghat River. And what we're going to do is scroll down to the bottom of the dashboard. And here at the bottom left, the occurrence data breakdown is shown in the occurrence data panel. So this shows you all the taxa that were recorded at these sites and gives you a quick breakdown of the number of occurrences of each taxon, origin, endemism, and conservation status. But if we click download as a CSV file, we're going to get a spreadsheet with all of those data records and quite a wide range of different attributes that that companion those data and really add a lot of value and insight into those data. And that can be viewed by clicking download as a CSV. All right, the same dialog pop-up comes up. And again, no specific use. This time we'll call it test 2. And once again, we'll hit download. And then you'll see a note comes up saying that your download request has been sent to our staff. So this is then verified by an FBIS staff member, and you will then receive an email with a link to download your CSV file as soon as it's ready. Now, this generally happens fairly quickly. It can take up to an hour or two, but generally you should receive that fairly quickly. All right. So next, I'd like to show you what the CSV file actually looks like. So now that I've downloaded that, I'm just going to open that up. And share that with you. All right. So this is what the CSV file looks like. It's very similar to an Excel spreadsheet, and it can actually be exported to an Excel spreadsheet quite easily as well. But for now, I'll just show you the CSV file itself. And like I said, this is really the raw data associated with all of the occurrence records in this data query. So each row is a data record, and each data record has its own unique identifier, which is this number here in the gray column on the left. And this is an automatically generated code for each record once it's been added to the FBIS platform, and it's a really nice way to trace an individual record. All right, so in this case, I'm just going to walk you through the main types of columns that are available and the main types of data attributes that you can then use to do further analysis on the data. All right, so after the unique identifier for the record, each record has its own original river name and river name. Original river name would be the river name given by the author. So if there was nothing specified, then automatically FBIS generates the river name based on its river's layer. In this case, the Rondeghat Rivier, as per the river's layer. But in other cases, like down here, you can see that in this, for these records, an original river name was specified as Rondeghat. All right, next to the site codes, once again, the original site code would be the name of the site given by the author, but then site code is an automatically generated unique code for each site once it's uploaded to the FBIS. If it's available, there's a site description column as well, which just helps you, I guess give some additional information where exactly the site is found. Then we have a column called refined geomorphological zone. Now, we also have a column called geomorphological zone, but just because of the layout of the CSV, it's a little bit further along, so we'll see that just now. But the refined geomorphological zone is really a column that would be entered by the user, if they don't agree with the auto-generated geomorphological zone that FBIS populates the data with, based on the geomorphological zone layer on FBIS, which in our experience generally does a pretty good job. So usually this is not really needed. Then next up, we have the spatial coordinates for the record, sampling date for the record, and then the basic taxonomy right the way through from kingdom down to taxon. Taxonomic rank is the finest taxonomic resolution for that record. For most of these fish records, it's species, but in other cases, it might be subspecies or it might be family for a lot of the invertebrate data. And then we have some information about the sampling. And this is, I think, really useful information where it's available. We have sampling method, and then we have the effort and then the catch. So it's really the breakdown of the catch per unit effort of, in this case, fish records. So we can see that for a lot of these data, there are no there is no information on the sampling method and the effort, but there are a few records down here at the bottom that have, maybe I'll just zoom in on this a little bit here, and I can show you that we have, in the sampling method column, you can see the method was a FICNet. In the effort column, you can see that the effort was time. In other cases, it might be area or some other measure. And then we have the sampling effort value. So you can see that in this case, it was FICNets that were set for 720 minutes. And then you can see the abundance measure of the catch. So in this case, 14 individuals of Ostroglanus scilli were caught in this FICNet that was set for 127 minutes. So really detailed catch per unit effort data available for the study, which is fantastic. But yeah. All right. Moving on, there's some information about the biotope where the record was collected. So you'll see for these data that's not available, but it's often more common for invertebrate data. And just to quickly go back to the abundance value, you know, in a lot of cases, it's just a record of a taxon at a place. So that's why these cells are blank. And the actual abundance of a taxon at a site is only indicated when that data has actually been provided. So I'll just let you know that all of these top rows that are blank are actually data from GBIF. And for most cases in GBIF, there are no abundance information. That's more data that has been uploaded from, for example, scientific publications or other data sets or reports to FBIS. Right. Then some more attributes about the taxon itself, whether it's native or non-native, what its endemism status is, what its conservation status is, both globally and nationally, and then onto some information about the source reference for the records. All right. So I've chosen this example specifically because I wanted to show you two different kinds of source references. So for the top rows here, you can see that these are all actually data that that the source is the Global Biodiversity Information Facility, GBIF, data that are pulled into the FBIS platform automatically from the GBIF platform. And for these GBIF data, there's a certain set of attributes that come down with those data. Importantly, we can see there's the collector owner and the institute of the collector and the owner as well. We know that this reference category is a database, and that's the title of the database. And we also have some important information about these records coming straight from GBIF. Importantly, the GBIF ID, the kind of record, whether it's a preserved specimen, for example, and then some other information about the institution, the catalog number for the record, and who it was recorded by as well. Then, if we look lower down here at these records down here, these were actually collected, these are not GBIF data. These are data collected by Jody Reisenberg, a student at the University of Cape Town. And there's some basic information about the authors of the study, the year of the study, and the source in this case is not GBIF, but it's an MSc thesis. The reference category is a published report, not a database. And then there's the title of that thesis of Jody's and a link to it as well. All these other GBIF attributes are obviously not relevant for the thesis, so those are all left blank. All right, so that really covers the source referencing. Then next up is a really important column, which is decision support tool. And this just indicates data that have been used, checked by experts and used previously in a decision support application. In this case, you can see that some of these data from GBIF were actually used in the 2022 DFFE screening tool. So it's a really nice way to track which data have already been verified and used for that purpose. All right, then we have record type, which is a new column that we've just added. And we're still busy populating that, but that really just shows whether the record is what kind of a record is it? Is it a DNA sample? Is it a preserved specimen? Is it just an observational record? Which it's also really nice to know what category that is. Right. Next up, we just have a whole range of environmental data or abiotic data or physicochemical data, depending on what you want to call them. But these are really the most common kinds of abiotic measurements that are collected alongside biological data when biological samples are collected. So we have things like water level and turbidity and embeddedness, and then a whole range of physicochemical factors, really quite comprehensive. And you can see that for most of these GBIF data, there are no abiotic companion data with those. But for Jody's thesis data down here, you can see that some of these cells are populated, which indicates that there were indeed some abiotic data associated with her biological fish records as well. All right, next up, we have a lot of spatial and environmental attributes associated with the fish occurrence records. And I'm not going to go through all of these one by one, but more just to let you know that what these really are columns that represent some of the key layers that are available in FBIS. For example, that this particular fish record was located within a particular river management unit, within a particular FIPA fish sanctuary, and within a particular system resilience category as well, and so on and so forth. Whether you're looking at thermal metrics, whether you're looking at catchment location, etc., that record will be basically has intersected with these other management and spatial and environmental layers on FBIS, including a wealth of thermal layers available on FBIS as well. So you can really get quite granular. For example, if you're interested in thermal information related to that data point, you can really take quite a deep dive into some of the monthly thermal information available for the site where that specific record was recorded. All right, everything through the management layers, like the water source area that the record was fallen within, the catchment that it occurred within, the hydrological region, several of the FIPA layers. Here we have the geomorphological zone, like I mentioned to you earlier. This is where the geomorphological zone of your record will be found, and more other management layers related to the present ecological state, critical biodiversity layers, some more thermal metrics, ecoregions, freshwater ecoregions, some more thermal metrics, and then finally just the GBIF key, which is a repeat column just right at the end for GBIF records, just in case. All right, so that's really an overview of that CSV file. And you can see that there are a lot of columns here, and don't be overwhelmed by that. What I would rather encourage you to do is to, once you download this CSV, you just know that you've got all of these associated columns to analyze with your biological data if you need them. And you can be quite selective in what you choose to include in further analyses by searching your spreadsheet for a particular column of interest. And just a final reminder that, once again, if you want to, you can export this file to a Microsoft Excel spreadsheet as well, if you prefer. All right, so that's it for tutorial 7. And the next tutorial, we're going to be focusing on adding a site to the FBIS platform. Thanks very much, and see you all next time.